Hi, everybody, and I want to first thank the organizers of the annual meeting for giving me this opportunity. So I'm going to be talking about clonal expansion of pre-existing lung cancer driver mutants induced by benzo fluoranthene. And Keith gave a great introduction of why we're looking at cancer driver mutations, and the focus is on non-genotoxic carcinogens, but this is the first application of CarcSeq to a genotoxic carcinogen, which I think is interesting. I also want to say that Stephanie Smith-Rowe is a co-author on this presentation because she has kindly allowed me to present some beautiful data that she has generated on mutagenesis, which provides a really nice context for the clonal expansion piece. Next slide, please. Okay, so Stephanie and I are participating in this amazing collaborative study on benzobifluoranthene that's being led by Carol Yauch, who's at University of Ottawa. Sorry, I didn't put that, Carol, and Francesco Marchetti of Health Canada. And what they've done is they have designed this experiment where mutamouse male mice are treated with daily with benzobifluoranthene, a mouse lung carcinogen. They've looked at 5 doses in vehicle control, 8 animals per group, and they've treated for different durations from 28 days out to 180 days. And a lot of interesting data is coming out of this, and it has really advantages of being able to intermesh it all. Standard genetic toxicology tests, but also error-corrected NGS approaches. And in the box there is what the results that I'll be presenting today. So Stephanie's group at NIEHS has performed duplex sequencing using the twin-strand mouse mutagenesis panel on mice treated for 28, 90, and 180 days. And here at NCTR, we've done carcseq analysis of mice treated for 90 and 180 days. And I'll also show you some histopathology that was done on the 180-day mice. And thanks again to Stephanie for letting me present her data. Next slide, please. So this is beautiful data. Again, it's duplex sequencing using the twin-strand mouse mutagenesis panel. And what you can see very clearly is that at each time point, there is significant dose-related increase in mutation frequency caused by this genotoxic carcinogen. What may not be obvious to everybody is there's 2 ways to calculate duplex sequencing mutagenesis data, and that is mutant fraction min and mutant fraction max. Mutant fraction min means if you observe the same mutation more than once, you only count it one time. And what these figures show is that there's minimal differences between mutant fraction min and mutant fraction max, suggesting that using the mouse mutagenesis panel, which is designed to capture neutral mutations, you see little to no clonal expansion of these mutations. Next slide, please. And this is the mutation spectra of the mutations that were recovered using the twin-strand mutagenesis panel. And the top left is the control animals, and the bottom, it increases in dose down and then to the right. So the bottom right has the highest dose. And this data was analyzed by Xinwen Zhang at NIEHS, Stephanie's postdoc. But what it shows is that there is significant induction of primarily C to A mutation, which fits what is expected for a PAH. Next slide, please. And I believe Stephanie's on the call, so if I get anything wrong, she can correct me. Okay, so let me give a very brief introduction to CarcSeq. It's a way to quantify clonal expansion of cancer driver mutations. So it's PCR based. The unique molecular identifier sequences are incorporated as part of the primers. And then error correction by consensus is based on construction of single-strand consensus sequences. We look at between 300 and 325,000 single-strand consensus sequences at every position of every sample. But because it's PCR single-strand consensus sequence based, it's not as sensitive as some other error correction approaches. So for this reason, we apply a cutoff of 10 to the -4 to reduce the potential impact of uncorrectable PCR errors that may occur in the first few cycles before UMIs are fixed at both ends of each molecule. Nevertheless, because we're looking at 300,000 single-strand consensus sequences and using this cutoff, that means that there are 30 more different UMI-defined mutants being recovered for every specific mutation that I'm going to show you. And I think using this higher cutoff is fit for the purpose of analysis of clonal expansion. Next slide, please. So this is the data, all the mutant fractions that were recovered in the lung DNA that we analyzed in the 180-day treatment group. So there were 6 mice per dose group. The doses were 0, 6.25, 12.5, and 25 mg per kg per day. You can see the mutations in the graph are color-coded by dose. They're arrayed in this graph on the x-axis by their panel position, what amplicon they occurred in, and on the y-axis by their mutant fraction. Number of mutant single-strand consensus sequences divided by the total number of single-strand consensus sequences recovered at a given position. And what you can see is the data sort of arrays up in columns that recurrent mutations are common, and we're recovering the same mutations in multiple mice. And the other thing that's clear from this is that most of the highest mutant fractions are coming from the 12.5 mg per kg treatment group. Next slide, click forward, please. Yeah, so you would expect it to be the high dose, right? 25 mg per kg would have the highest mutant fraction if it's clonal expansion. But in fact, the 25 mg per kg treatment group exceeded the maximum tolerated dose. So those mice showed a 16% lower terminal body weight than controls. And it's known that exceeding the maximum tolerated dose is known to delay tumor development. So I think it's plausible to consider that it might also delay clonal expansion. Next slide, please. So this is the main important finding from the study. So we have been investigating a metric of clonal expansion called median absolute deviation in mutant fraction. You might imagine that as clonal expansion occurs, the variability in the mutant fractions recovered in a particular mouse are going to increase. And that's what MAD and mutant fraction is. It's a metric of variability. And each one of those dots represents a different mouse. And what you see is that when we calculate MAD and mutant fraction from what we believe are the lung-specific amplicons and drivers, we see a dose-dependent increase in MAD and mutant fraction with BBF. It goes up to 12.5 and then down at 25. And there was a significant pairwise comparison between the control and 12.5 mg per kg dose. If you calculate MAD and mutant fraction from the other panel sequences, the ones not considered lung-specific drivers, you don't see any significant dose response. And we also saw a significant dose response in terms of just recovery of epidermal growth factor receptor mutant fraction, again, showing that significant pairwise comparison between the control and the 12.5 mg per kg treatment group. Next slide, please. Just click. Unfortunately, we looked at the 90-day treatment and we do not see a significant effect, whether we look at the lung-specific, significant dose response, whether we look at the lung-specific drivers or other panel sequences. Next slide, please. This is the normalized trinucleotide mutation spectra from the mutations recovered in mice treated for 180 days. It's stratified by lung-specific drivers on the left and non-lung drivers non-lung sequences on the right. And the control is on the top and the high dose is on the bottom. And what you see is that these spectra look like spontaneous and that there is no impact of dose on these mutations. For that reason, I think these results are consistent, in conjunction with the earlier result where I showed you the increases. This result is consistent with clonal expansion of spontaneously occurring or pre-existing mutations. Next slide, please. So the last data I'll show you is the histopathology that was performed at Hoffmann-La Roche, and this was conducted by Jürgen Funk, Andreas Zeller, and Sabrina Ken. And they reported to us very subtle bronchiolo-alveolar hyperplasia in a few animals that would most likely not have been considered test item related if the mode of action of the compound were not known. And those data are shown in the table at the bottom left. So no tumors were observed with BBF treatment, and 1 of 3 ENU positive control mice had a lung adenoma. On the bottom right, what you see is concordance between our MAD and mutant fraction measurements and the percentage of mice with bronchioalveolar hyperplasia. And you see the same effect about the highest level of mutation in the lesions in the 12.5 mg/kg and not the 25. Next slide, please. So to summarize, BBF is a genotoxic mouse lung carcinogen. It caused dose-dependent induction primarily of C to A mutation observed by duplex sequencing, and that's Stephanie Smith-Rowe's beautiful work and her collaborators. We saw clonal expansion of cancer driver mutations in lung samples treated for 180 days that was concordant with histopathology. It's known that BBF activates both AHR and CAR receptors. So through either a mutagenic mechanism involving clonal cooperation or a mechanism involved with receptor binding and activation and altered chemical activation, BBF causes clonal expansion of pre-existing cancer driver mutations that are presumed to progress BBF-induced lung carcinogenesis. And there's a lot of people contributed to this work, and they're listed there, but I specifically want to mention Jennifer Foskey here at NCTR, who did all the carc-seq analyses. I don't know if there's time for questions, but if there isn't, I'll be happy to try and answer things in the chat.
Thanks. Many thanks, Barbara. I think we could do one question if anyone has one. Feel free to come off mute or to raise your hand. We see some claps for you. I think it's a really nice study. It's another really great example of, utilizing people from different organizations and sectors to get the work done. And I think using the study done from Carol and Francesco's lab, showing the different applications.
It really has been great to be a part of that. So I thank Carol and all of those collaborators too.
Okay. I'm not seeing anything, but maybe we'll take you up on the offer. If there are anything, we can continue to have in the chat. Thank you very much, Barbara.
Thank you.